Computational Biology Department at Sloan Kettering Cancer Center. I'm going to be presenting on our work on network synergy screen and network modeling, or sorry, drug synergy screen and network modeling in liposarcoma. So we work in the field of cancer systems biology, and in this field we're really taking advantage of all the progress that has been done in these other areas indicated here in therapeutics, the development of targeted drugs that target specific alterations in genes that are drivers in cancers, and in genomics from the tremendous efforts on really mapping out all the genomic drivers in different cancers. And on the proteomic side, we take advantage of the developments, particularly in mass spectrometry, to map out recently the entire human proteome of different tissues, but also the phosphoproteome, because this really characterizes some of the changes in the signaling pathways that are altered in different cancers. So the challenges that we're faced with in cancer systems biology are to develop cancer-specific signaling pathway models that take these specific genomic alterations into account, so we can start designing drug combinations and making models that also take both tumor intrinsic factors and extrinsic factors into account, including tumor stroma interactions. And with this, we hope that we can help to help out with some of the challenges that are in therapeutics, for instance, the problems there are with drug resistance to targeted drugs, by suggesting combination therapy, also helping out with predicting and characterizing the genomic alterations in different cancers, so we can take into account tumor heterogeneity. So our take on this is, compared to the previous talker, is much more small scale, but where we're doing experimental perturbations to the systems in order to derive context-specific pathway models. So this is really our take on how to do cancer systems biology in order to model the underlying changes in the signaling pathways that we can then use to predict, for instance, new drug combination therapies. So our typical workflow is that we work with targeted drugs and do perturbations in all combinations, and then do data-rich profiling of the cellular response, and that's typically measuring changes in phosphoproteins. And from that, we can then use our computational network inference models to derive interactions directly from the data, and this can also be assisted by prior knowledge incorporation. What we're using it for is both to describe the underlying pathways and have a model of the underlying pathways that are altered in specific cancers, and then we can use this to predict beyond the training set new combinations of drugs that we can rank and validate further in clinical settings. So it is an iterative approach that we're taking here, and we've recently published our sort of what do you call it, like a cookbook for this, where we were working with melanoma, BRAF mutant melanoma to predict new drug combination therapy for overcoming the resistance to BRAF inhibition. But what I'm going to be talking about here is liposarcoma. But before I go into that, I just wanted to have one slide on our network inference framework. So as I said before, we're using the drug experimental data that we've collected from the set of cell lines that we're working with, the phosphoproteomic readout, and we can use prior knowledge as well. This is optional in that it can be rejected in the modeling system. But we have a set of coupled ordinary differential equations that we describe this system with, and then the task is to infer these interaction parameters, the Wij matrix. And here you can see this for a toy system that we infer. Most of the interactions that we infer are non-interactions, or the probability distribution is centered around 0. And then we also infer some positive interactions and negative interactions in this way. And by describing and having the system as a set of probabilistic values for each of these interaction parameters, then we can now draw a set of good models that fit with the experimental data. And the prior knowledge in this case is encoded as a bias to these probability distributions, and if it doesn't fit with the data, it's rejected through our error function. So we can do two things with our network inference framework. We can either just describe the underlying pathway in a qualitative sense, so more the typical textbook pathway models that you see, but more importantly, we can draw a set of good models and use these for prediction, because we now have a set of coupled differential equations. So for the liposarcoma network, what we did was we had a few goals in mind working with this disease that I'll get back to in a second. But the goals were to identify synergistic drug combinations for potential treatment of liposarcoma, and also infer disease-specific network models that we can use to simulate the altered signaling pathways. And the questions we were trying to address were if we could use, if these network models could even capture drug synergy, and also if they could we then describe some underlying pathway mechanisms to observed drug synergies. So we chose to work with dedifferentiated liposarcoma, or DDLS. This is a very rare tumor of the soft tissue that arises from fat cells, and there are no real good targeted treatments to this disease. So as you see here, there are some areas that are well differentiated, where you can see the adipocytes, and then there's these sharp transitions into very dedifferentiated areas with lots of stromal infiltration. And these tumors, they present as large tumors either in the extremities or in the peritoneal cavity. And the treatment is mostly surgery, radiotherapy, cytotoxic drugs, but despite that, there's a high recurrence rate, and when the disease comes back, it's even more metastatic. So at Sloan Kettering, we are doing clinical development of targeted drugs with the CDK4 inhibitor PD991, palbociclib, and that shows prolonged progression-free survival. And this drug is particularly effective because in DDLS, almost all of the patients have an amplicon at 12q with CDK4 and MDM2 amplified. There are also other mutations in other driver genes, and there's a very complex general copy number alterations in DDLS. And here we're working with the clinicians at Sloan Kettering that have derived cell lines from patient samples. So here we're working with a cell line DDLS 8817 that was derived from a patient, and here we're doing array CGH to map the copy number alterations, and in orange you see a set of different tumor samples, and in blue you see the specific cell line that we chose to work with. And this cell line has some of the characteristics of genomic alterations, including this 12q amplification. But besides that, you can see that there's quite a lot of varying deletions in blue and amplifications in red. So we thought that this cell line is a pretty relevant model for DDLS, although with all the caveats of using cell lines to model human tumors. We then decided to select a median set of drugs, 14 drugs in all targeted to canonical pathways such as AKT pathway and mTOR pathway, and also some HDAC inhibitors. And these drugs are in preclinical or clinical trials. So to determine whether we have drug synergy, we did some experiments, and our framework for determining synergy is just based on the low additivity, and that simply says if you have two drugs, A and B, and you've identified the concentration that inhibits 50% of the cells, then you can ask the question, what happens if you combine these drugs? And it's an additive response if you get the same effect but at half the concentration of the single drugs, whereas it's synergistic if it's a larger effect and antagonistic a smaller effect than expected. So what we did in the lab is we set up several cell viability experiments using the Rosazerin assay and simply doing dose response curves, so increasing the concentrations of drugs and then calculating, in this case, the IC50 of each single drug and also the IC50 of the combined drug. And in this way, we had a very simple calculation to determine the drug synergy using the drug synergy index score. So a score below 1 is synergistic, above 1 antagonistic in this case. So here are all the collected results for the 14 drugs that we tested in this cell line. In green, you see most of the combinations are additive as expected. It fits very well with the literature that around only 10 to 20% of drug combinations are actually nonlinear or epistatic effects. In blue, you can see some of the antagonistic combinations, and red, synergistic combinations. So examples of the synergistic drug combinations that we identify are these parallel pathway inhibitions of MAPK pathway and PI3 kinase pathway, or dual receptor tyrosine kinase inhibition with MET and EGFR. And that fits with the theory behind synergy, that you often need to inhibit parallel pathways to obtain a response that's greater than additive, and also what the previous speaker referred to. One particular combination that we were interested in, and that our clinical partners were also very interested in, was this increased or synergistic effect of inhibiting IGF1R and CDK4. So as I told before, CDK4 is one of the primary oncogenic drivers in DDLS, and we're doing the clinical development of this drug. IGF1R is a less obvious target. It is in clinical development for other sarcoma subtypes, Ewing sarcoma, but it's really a non-trivial combination that we discovered this way because IGF1R is only altered in 3% of the patients. So this combination would not have been identified if you just did genomic screening. So we chose to work further with this drug combination and mine further into what are the mechanisms of drug synergy. But first, we just wanted to confirm if we had a true synergy or not, and we used a couple of different cell lines. Additionally, more than shown here, and we consistently find that this combo is synergistic. We also tested with different drugs targeted to the same molecules, and also siRNAs to these specific targets. So that confirms that we have a synergistic drug combination, but what we didn't know was how do this drug combination actually work on a mechanistic level, and here we thought that our network modeling approach would be one way to deduce that. So then we set out to do a series of experiments where we did the same drug combinations as before, but now instead of measuring cell viability, we measured the changes in phosphoproteins based on reverse phase protein arrays that I'll come back to. And then we wanted to use our network inference approach to see if we could describe the underlying mechanism of this observed CDK4 and IGF1R synergy. So we used reverse phase protein arrays mainly for practical reasons, because it's very high throughput on the sample side. We're now venturing in and doing a lot more mass spectrometry, but this is still not very high throughput on the sample side. So here it's very easy to have all your hundreds of conditions and take the cell lysate and spot it onto these chips, and then each chip is incubated with a different antibody. So it's very high throughput on the sample side, but the problem is, of course, you rely on antibody specificity, and it's not like in ELISA where you're using 2 antibodies. Here you're only using 1 antibody. So we did the experiments, all the combinations seen here of these 14 drugs. It just choosing one dose that was based off of Western blot experiments to find a dose that inhibited the target at sort of IC40 level, if we could get that. And here is the normalized output of these screens with the reverse phase protein arrays, where green indicates an upregulation of these proteins or phosphoproteins, and blue indicates downregulation. So although we had a set of around 100 different antibodies that we measured, we narrowed it down to around 15 antibodies that showed good correlation between our biological replicates and that were involved from prior knowledge in DDLS mechanisms, and were some of these some of these points that really capture the canonical pathways. So with that data in hand, we also included the cell viability measurements, as I showed you earlier, and we used some background pathway knowledge of interactions that have been described elsewhere in the literature that can also be incorporated in our modeling approach. So we ran our network inference approach, and then here is a sum of overall the interaction parameters. So this is really an aggregate of all the possible solutions that there are in this space. So we don't typically try to interpret these still pictures too much, because as I said, it can be multiple solution states. But we do recapitulate some of the canonical pathways from MEC down to ERK and P76K and S6K and cell viability. But what we instead want to do is use a set of these models to predict a response to drugs. So that's what we did. We had the network models, and we could now take two different nodes and then increase the perturbation strength to these nodes. Or in other words, deactivate these nodes in silico, and then have a measurement of the predicted cell viability node. And that's another point of our pathway modeling approach, that we can combine viability measurements and molecular measurements into one model. So having that, we can then, again, similar to before, calculate the synergy over model. And here indicated by yellow is an effect that's greater than expected from just the single perturbations. So aggregating all that data, doing all drug inhibitions in this network, we then get the following distribution of synergy scores, where here are the antagonistic ones and the synergistic ones in red again. And we do recapitulate several of our experimental drug combinations, including IGF1R and CDK4. And this is actually pretty non-trivial, because we're predicting based on molecular readouts coupled to the phenotypic readout. And in the molecular readout, we only used one concentration. So I think it's quite powerful that we can simulate what is going on a network level, and then predict what the effect is on cell viability. And this was, of course, done using a cross-validation procedure, so we're not predicting and training on the same data. So next, digging more into the CDK4 and IGF1R synergy, what we did was we removed one edge from the network at a time, and then recomputed the synergy. And then here are the edges that indicate when we have observed a decrease in synergy score. So in other words, edges that are important for mediating the drug synergy. So what we see is, naturally, when we take away the edges between CDK4 to cell viability or IGF1R to cell viability, we destroy the predicted synergy as expected. But then also we predict that EGFR to AKT and further down is potentially involved in mediating the drug synergy, whereas other pathways such as MEC to ERK is not predicted to have an effect. So I'm running a bit out of time here, but what we then went on to show was that this fits also with the experimental data. So we went back to the lab, did some Western blotting, and basically what you can see is that when we inhibit with CDK4 and IGF1R, in the combination treatment, we see a much greater effect on downregulation of phospho-AKT than the single drugs alone. And similar to what I said before, there's no effect on the dual perturbation on phospho-ERK level. So it seems that the network model captured some of the mechanisms of drug synergy, although of course you always need to verify experimentally whether your predictions hold up to scrutiny. We also combined AKT and CDK4 just in vitro in different cell lines, and also observed a synergy on a phenotypic level. So in summary, what I hope to shown is that we have this medium scale systems biology approach where we're really interrogating the system that we're working with through drug combinations, and developing context specific pathway models that can be used to predict mechanisms of drug sensitivity or resistance. In this case, we were working with DDLS, and we identified the CDK4 and IGF1R combo that we're testing in clinical settings at the moment. There's still a lot of work into figuring out exactly how the AKT pathway is involved, because there's a non-trivial connection between inhibiting CDK4 and actually getting an increased effect on phospho-AKT with IGF1R perturbation. So we're looking further into that at the moment. Finally, I want to acknowledge Evan, who worked a lot with me on the network modeling part. And these guys in the middle here all worked a lot on the experimental settings, and our clinical collaborators are Sam Singer and Gary Swartz at Sloan Kettering, and my mentor Chris Sander for great guidance. And thanks for your attention.
So you mentioned that in building your networks, you used a framework which involves ODEs. Does this mean you need to generate time series data from the RPPA array?
We assume a steady state, so in principle we can use time perturbation data, and we're doing that now, but in this experiment we didn't have enough throughput to do it, but we're doing that at the moment.
Can you comment a bit on how concentration dependent these synergistic or antagonistic behaviors are? Because your starting concentration could determine if you start at a really high concentration, and if it reaches saturation, then if you add in this other one, you may just observe antagonism. And I was just looking at your kind of dose matrix, and it was actually showing synergism. Maybe it's not reached saturation, so some comments on that.
Yeah, that's a very good point. So we didn't do the dose matrix for all combinations due to limitations in the experiments, but for the follow-ups, it's certainly true that you can have these fields of where there's some unexpected interaction between low dose, high dose, etc. We didn't see any pockets of like supra synergy or sub synergy effects, but other people have been reporting these types of non-trivial interactions.
But would you say that at very high dose combinations that you would expect to see antagonism, or is that not what you would intuitively?
Yeah, so another thing related to your question is that at least for the calculation of synergy, although I showed that we did it at IC50, we actually can do it at any level we want and calculate the synergy, and definitely you can't do it at IC20, then you will not see the synergy. So yeah, if you have too high concentrations, you can't observe synergy. Yeah.
Hi, thank you for the talk. I have two sort of related questions. One, did you have any false positives in your model or false negatives? And two, are you interested at all in looking at the predicted antagonizers, because that could also be medically relevant? Yeah,
so yeah, we didn't dig further into the predicted interactions, so we had a lot of new predicted connections, and that's definitely something that always should be done with these types of experiments, but But again, capacity, it's hard to really say, do we have any true negative predictions and true positives? So we were relying a bit on only following the true positive story, but in principle, we can also follow other stories. And about the drug antagonism is, of course, equally interesting. And what we're working on now is on a molecular level, do all these dose matrix perturbations where we are exploring the whole field of concentrations and then seeing on a molecular level if you get these antagonism response as well.
Thank you.
Thanks.
I guess my question is pretty related to the previous one, but my question is regarding the bottom line, I guess. So I've observed a number of drug synergies or antagonisms, and you've generated a model to predict, to explain those synergies. Can you go in reverse and actually count the times that a model would predict synergy and then you actually observe synergy? Do you know what that percentage is? What's the accuracy in terms of getting the right calls?
Yeah, that was not tested, so I can't give you a specific answer to that.
Right, but you can imagine how that's a very important question in the field, right? You've given primary data, can you predict synergy? And it seems like you're in a great position to sort of answer that question, the degree to which we could predict synergy. And you have sets of 14 by 14 drugs to answer that question.
At least we have the experiments there, and when we train and hold out data and then predict, then we can actually define a set of true positives and true negatives in terms of drug synergy. So yes, we can do that. Our limitations with this project was that we weren't 100% accurate in predicting all the drug synergies that were also experimentally synergistic. But one of the main limitations was that I mentioned shortly, is that we really only used one drug concentration for the molecular readouts, where we would need more drug concentrations to effectively capture what is drug synergy and not. Yeah.
Hi, so it's nice that you could sort of show that the predicted synergisms corresponded to the experimental ones in this case, but what was the overall correlation between predicted and measured?
So we had I don't remember the R squared, but we had a linear correlation between the CI score, the combination index score, and the computed synergy score. But again, it wasn't as strong as we had hoped it was, but I must say again, you're just repeating myself, that the limitations were on the number of that we didn't explore different concentrations thoroughly enough.
You don't remember the correlation coefficient?
No, I don't, but it was positive and significant, but.
Just a quick question. At what scale can you screen these pairwise drug combinations?
Yeah, so that's a good point. We were doing this by hand, but of course you can scale up with robotics. And what we're doing now is also taking global approaches such as pooled shRNA screen for a little different purpose, but we would like to increase the throughput on the drug condition side as well, because we're worried that we're under-parameterizing our models. Yeah. Thanks.